based out of the Flat-Lake Biological Station. He received his bachelor's from Iowa State University before going on to do both his master's and PhD at the University of Montana. And he hasn't just stuck around at Montana the entire time. He went off after that, spent some time at Joseph Fourier University in Grenoble, France, before getting a position at the CRNS in France, which is similar to the USGS. Spent some time then at University of Porto in Portugal before coming back to first professor position at University of Montana. His research focused on conservation and population genomics, or uses genomics to inform conservation actions and decision-making for imperiled species. Also done a bunch of work recently on detecting invasions using eDNA, and talked about a bunch of the new exciting genetic and genomics tools that we're using in ecology and conservation. Was talked yesterday. So today it's going to be kind of an informal kind of Q&A. We have a list of questions that were posted online before, but these are a lot more exciting if there's a lot of interaction with the crowd. So if you have a question, raise your hand. Get my attention and call out as well. So I'll be looking around for arms up. I'll also check for Zoom buttons throughout. Yeah. So does anyone have any burning questions that they really want to start off with? Otherwise, we'll go to the sheet. Oh, we are on the back.
Sorry, could you repeat, please?
What
Yeah, so I chose to work in conservation and genetics because I grew up loving hunting and fishing and hiking and boating, camping. And then as an undergrad, I took a genetics class and liked it. And then I discovered someone had combined the use of genetics with wildlife management. And I didn't realize, and it didn't actually exist as a discipline. And now there's a couple textbooks on conservation genetics. So I realized I could potentially combine genetics with conservation, and I drove out to Montana. And long story short, I found a professor who was interested in being my mentor, supervisor, accepting me as a master's student to use genetics on an exciting bighorn sheep project. And from then, there was no looking back. Just love it. I think a lot of biologists get into biology because they love the outdoors, you know. I haven't hunted for years, but my motto in high school was, if it flies, it dies. But I don't know.
Along the similar lines, was there a particular course in undergrad that really kind of moved you down that path toward?
Yeah, actually, I discovered conservation genetics because there was one paper in this grad. So I was an undergrad taking grad seminar. And we read this paper talking about how you could use genetics for conservation. Yeah, it turned out I worked for the guy who wrote the paper eventually, which was helpful.
Okay, no other questions from the room right now. We'll jump into the third ones. There's a number of categories here. There's even options to choose from if you'd like. Pick from origins, secrets of success, research related, or broad other topics.
Secrets of success.
Secrets of success. All right. So in your work, how have you in the past identified new avenues of research as you've been moving through your career?
So I would say, and it changes throughout your career, but literature review, to see what the publications say are important questions. So find an important question. To find out what the important questions are, you need to know the literature, but then you network with folks, you talk with them. And important, you know, I would say has three components. It has to be what you love and what excites you. It has to be, I would say, important to both basic, if possible, basic research, basic understanding, and applied, so that eventually you can draw on both those funding streams, or advanced basic understanding and have it be applied. Yeah, I guess that's the main. In other ways, so reviews, talking to people. Oh, calls for proposals. I was soft money for 5 years, at least, so I needed to raise my own salary. So you sort of have to follow the money to some extent. So you look at calls for proposals. And then another secret in there I discovered was you can influence the calls for proposals if you talk to the agency that's developing the call. It might be hard to get in that position, but they might come to you if you've published in the area and ask you if you'll help them write a white paper, which is to help develop their next call for funding. So that was an eye-opening when I learned you could be involved in advising an agency like NASA, what they should spend money on in the next 10 years.
So it seems like a great way to inform, you know, what the important questions actually are that we funded in the future.
I have a follow-up to that. So did you tell them that they should put their money in exactly the areas where you were interested? Or were you thinking more?
I had tried to be really fair and open and broad, but I did feel strongly and realize that NASA in its ability to remotely sense environmental variables across landscapes and waterscapes hadn't used genetics to characterize things in these pixels across landscapes. And so I had suggested that they combined their massive big data source with the exploding massive big data source of genomics, particularly landscape genetics, so that you could then use environmental variables to help model and predict movement of organisms across landscapes. And they were already interested in it, but I think the first proposal I submitted to them to do that, they hadn't realized it was a thing. So the next call came out and it included that, which was cool to see. But I think that happens more than we realize. That's like one of maybe a couple times it's happened with me. But if you work with state agencies and you have an established long-term relationship, especially like trust, and you're never, you know, sort of perceived as overselling them on something like geneticists have done. Oh yeah, new non-invasive sampling, eDNA can tell you everything you need to know. You don't want to get caught overselling. But if you have a relationship and some trust built, you could help them decide what to do with their money and get them data they need to address their questions. And thus, they'll either have an open call. Most agencies have to have open call to everybody.
I guess what, kind of keeping along similar lines, what are some of the kind of big shifts you've taken in your kind of focus of your research over the years? So I know you've worked on a number of different species through time. The question of what are some of those shifts you've taken and what sort of drove you to those?
So at first, I just wanted to work on large mammals and conservation. So I did that, and then to be able to do that, I took a postdoc on domestic ungulates in Europe, which is like my dream postdoc because I had to travel the world literally and sample rare endangered breeds to characterize them genetically. So while I was in Mongolia and in Egypt and other countries, I made a way to get samples from the trophy wild ungulates, or so I transitioned into domestics and learned a whole new area of research and discovered the wild progenitor of all domestic goats and published that sort of thing. And then I wanted to get back to the US. So while I was in France, I guess, I don't know, I shifted. What would I say? I guess I got into aquatic research more when a job opened up at the field station on a big lake where they needed help with the early detection of invasive species. And I was sort of pre-adapted to that because I'd done ancient DNA on goat fossils, you know, so I could get DNA out of anything or most anything. And it's just an important problem. One of our nation's biggest, I mean, I'm biased, of course, ecological and economic problems is zebra mussels coming in and costing hundreds of millions of dollars to the region they invade. So wow, I attacked that with basic. It's a lot easier to get DNA out of water than a fossil. So I shifted to aquatic, And then I needed funding as a soft money person, and I realized my advisor and others had told me all along, that fish are one of the best model species because there's hatcheries and there's money. And you can ask her questions, it's not quite mice or Drosophila, but fish is just a great study organism. And genetics can really help, about anything, but they can really help with addressing questions fish biologists need. So then I addressed the question that my main mentor had addressed all his career. What's the effect of hybridization with non-native invasive fish on the local native fish? And still working on that a bit. So that was a shift to fish, in part because I needed money, but it's also a great research system and had collaborators. In the state I was working, wanted to know some answers. So yeah, I'm more aquatic and fish focused nowadays.
That's the right direction. Feel the move is towards fish, in my opinion.
I knew you'd like that.
Yeah.
But would I go to Drosophila? I don't think so. I talked with Brian and others about the beetles. That sounds like a really nice system. But
Yeah, that was an interesting conversation yesterday. Hear about that. So next question under secrets of success. What kind of roles have mentors played in your success? Or how have your mentors helped you through different stages of your career?
So I've been really fortunate to have amazing mentors. And I partially kind of stumbled into it, dumb luck. But I would say the most important thing for success of grad students and maybe beyond is to do what you love, but then have a good mentor who supports you. And I hadn't realized how much support I had until I was out. And then a few years later, I got to read one of his letters of recommendation. And then I talked to other grad students, and he's just really supportive. And so he was just a really good scientist. I mean, National Academy level. And I was lucky because he's a really good person, not a big ego. And he loved to have grad students. So I just got lucky. But what you should do is interview grad students of the lab PI that you're going to work with. And then I was really fortunate to have two mentors. Try to have 2 or 3. You might need 2 or 3 letters of recommendation, but really what you benefit from is these 2 or 3 levels of different complementary expertise. So I worked with one of the best field ecologists in large mammals ever. And then I worked with what turns out to be one of the best, the grandfather of conservation genetics. And it wasn't by choice. I was going to go to Montana State University in Bozeman. And when I was sort of there visiting, someone said, why don't you drive 3 more hours to the University of Montana down the road in Missoula and go talk to the god of electrophoresis? And I'm like, what's that? Oh, it's a way to get genetic data on wild populations. So I drove down the road and ended up going to the University of Montana. So I hope that's helpful. I was lucky, but you should be more strategic in your, I mean, I was doing what I love. So I lucked out by having these 2 mentors, really complimentary and supportive.
What are some of the obstacles or setbacks you faced in your career, and how'd you overcome them? Along the same lines, is there anything you would have done differently to overcome them more effectively or learn better from them if you could do it again?
Obstacles and setbacks. I would say I failed to get funded repeatedly, and then I had some papers I failed to get published. So the key is persistence, and it's way easier to be persistent if you're doing what you love and if you have collaborators, good collaborators. So I was really fortunate to get a Fulbright grant as a PhD student to go to Australia, but the first time I failed and the second time I failed. And then I had help from a local mentor who connected me with a good lab in Australia who wrote a better letter of recommendation. So persistence is the key. I finally got that. And same with other grants. You can't expect at certain agencies like NSF, which stands for non-sufficient funding, you can't expect to get funded on your first round because they get, I've been on a panel there and we got 120 proposals and 5 or 6 were funded. So you got to resubmit, and I think the most successful year of submission is your third. So you got to plan to submit 3 times, at least, and then it's a crapshoot because. So be persistent and then in papers, I've had a couple papers that were shockingly rejected repeatedly because I knew it was good stuff. And it turns out there's someone out there who's trying to publish the same thing. And then I met with the guy eventually and talked it over and said, yeah, if you entertain this other alternative hypothesis, maybe it's publishable. And then it got rejected again. So then I went back to the original journal, a really good journal. And I told him not to have this guy as a reviewer. They loved it. They got published. So be persistent, but yeah, be persistent in publishing and in grant writing. And do something differently was another question.
Yeah, if you, was there anything you do differently about these obstacles in the past? Or is persistence kind of covered?
I would say, so early on, I had contributed to some work and I was a bit shy, maybe humble, and didn't understand the publication process. So maybe I should have tried a little harder to be invited to be a co-author. That's a little self-serving, but you have to be a little bit. So then I was a little more assertive the rest of my career and said, hey, I could help out with that if you need help, or, I love that topic. So, because publications matter, of course, in academia.
Follow up, you mentioned a paper getting shockingly rejected because you knew it was good stuff.
I mean, I'm pretty biased, of course, right?
Yeah, but how do you know? I think for a lot of students, they don't have the confidence in their own evaluation of
Yeah.
paper. How should they know it's good?
Yeah, thanks for following up. This is really important because I've had colleagues as well. I had another paper that was surprising. It doesn't, so if you get some really good reviews, I mean, they're really good, or they're even like, they're just really good. And then you get one that's not, and then that happens again. And you have co-authors maybe, and you talk to them. And by the time you're a postdoc, so this was an early postdoc, I just felt it should be published because I was getting good reviews. And maybe it wasn't until I discovered it was the same reviewer, more or less. And then it happened again, and then I talked to other people. So there was this one review I sent with some really good co-authors. We felt it was good. It coined the term landscape genetics. And one of the reviews came back and said, I'll never forget this. These guys got to be kidding themselves if they think they've stumbled across something novel. And then there was other personal attacks. And so it's like, that was so easy to respond to because it was so negative and personal that we just told the editor that, see these phrases, these are unreasonable, over the top. And the other reviews were really good. And then I have colleagues who also, this doesn't have this sort of personal attack or really extreme negativity doesn't happen much anymore, I hope, because journals, when they send papers to reviewers, send instructions to not do that, to be constructive and kind and give them advice. So you got to be persistent and not take it personal. And when you read that rejection letter, don't do anything for a day, read it again, and then get advice from your co-authors. Because often a rejection with possibility of resubmission is a really good thing, because they just want you to improve the paper and resubmit it improved. Because if you resubmit it, the time to publication is shorter than if you hold on it. So that was a strategy, still a strategy, I think, but 10 to 15 years ago, this reject invite became a big thing with molecular ecology and some other journals. So the point is, be persistent, try not to take it personal, don't overreact, and, talk with your colleagues. Do you have any other things to add? Anyone who's published?
Well, I was thinking, I had a postdoc right now trying to get a paper published that we both thought was really good, and he didn't have he had bad luck with to produce. And so I was asking myself, why am I so sure that this is good? And I guess part of it was, so part of it was, he had presented it at a couple of different conferences and gotten all kinds of great feedback after, like people coming out of the woodwork and modeling them the next day because they wanted to talk to them. It's like, okay, if we're getting that kind of response, there's something here. I think part of it is like what you said, you can get opinions from other people for more than this reviewers help you. But I think there's something intrinsic. Like, there's something, even if it's just you, who's only your opinion. You have a sense of what some of your work's better than others. Like, more important.
Yeah, and I would say if you can articulate that strategically in a well-polished response to the editor, I've been involved in a couple papers where they were rejected. And one of them I just knew better. So I helped the PhD student respond and they said, oh yeah, I agree now. You have two replicate populations that found the same result, and that is replication. And the main rejection of that was there's no replication there. But so that got accepted. And then on another paper, there was an author who's more senior than me and more experienced, and we chatted and he said, yeah, these reviews are off. So let's write a letter to the editor of genetics and explain to them why what the reviewer said is not true and why they should accept the paper. And that was accepted. So both those times, because we thought about it, we talked among authors, we were able to get it into that journal because we could respond well to the criticisms that were the so-called fatal flaws.
I think that being willing to respond to the associate editor with why you disagree with the reviews can go a long way. We just recently had a paper where the reviews were good, but the associate editor didn't like it. We were able to basically rebut their arguments and say why our analyses were appropriate, and they accepted it basically immediately upon getting that thoughtful response to their rejection. So there's a lot of things that folks are hesitant to argue against the decisions, but I think you can actually get a lot of benefit out of that.
Yeah, and some of the reasons that the higher profile journals for rejections is it's not novel.
Yeah.
So if you can explain in the rebuttal, you should do it in the cover letter in the article itself, but why it actually is novel, and then sufficient, it can really help to get the paper accepted. So I had with a postdoc a really nice novel paper, but it was on a topic that was really difficult to address. So what's the genomic basis? What's the effect of the I'll just cut to the chase. It was rejected. Like, this is the most rejections I've ever seen, but the postdoc was really persistent, and on the 7th or 8th submission it was finally accepted, and all that happened in a year and a half or so. So the resubmissions were like fast. And there was only one review of the 7 journals it went to that suggested substantial revision. So there was some math added to it. There was a lot of simulations, but some analytic equations were added, and it gave the same sort of result that a certain, if there's a trait that's adaptive and there's a major allele affecting it instead of lots of minor alleles only, it affects the population persistence. And that's quantitative genetics, and it's related to the environment. Heritability changes through time, and this hadn't been looked at before, and he looked at it. It was almost too novel and too complicated to be accepted. But after multiple, 7 or 8 times and a little bit more math, and then communicating with the new journal author straight ahead saying, yeah, this has been submitted 7 times. It's really polished, and we think you should accept it because of this, that the journal editor then set it up for review, and it finally got accepted.
Is it 7 times with the same journal?
No.
Okay.
That's what's shocking. Thanks for asking that question, because that's a lot of work.
Yeah.
Different journals, reformatting.
Yeah. And that's very persistent to keep sticking with that.
Yeah, and it's easy to not be persistent. And I have a couple of papers that I should have published that too late now. I just let them go.
Questions from the room before we move on to the next section?
Can you talk a little bit more about why you decided to do the postdoc and what the advantages and disadvantages for the work you brought?
Yeah, good question. Thanks. So I love travel. I had been to France before, and things just aligned. This conference was in my area, the International Bear Conference. And this guy showed up and talked about non-invasive sampling. How can you could get DNA out of hair and scat, and it hadn't been done. And I went to his talk, and I just loved it. And then so I had been to France on a undergrad study abroad thing. So I wanted to go back, and then I wanted to work on mountain ungulates, and I wanted to work on non-invasive sampling. And I just lucked out that the guy had funding. And they let an American accept European funding. It wasn't easy, but so I wanted to travel. And I wanted to do this kind of research, and I found a guy who had just advanced the field in a major way. He just established non-invasive sampling, get DNA out of scat and hairs. And so I just connected with him and stayed in touch. I was a bit persistent. And I got this email, hey, there's this funding, but oh, by the way, it's to work on domestic animals, not wild. Then I chatted with him, and we worked out a way to work on wild as well as domestic. So that's why I went overseas. And I would highly recommend, we were talking about this earlier, that your mentors, generally mentors tell their students to go to different institutes to broaden your perspectives. Don't stay at the same place for your master's or PhD that you did your undergrad or your master's. So I would recommend going overseas to another continent if you really want to broaden your horizons. And France is you know, it's got a tradition of being a world center for population genetics. And so that was helpful for me too, because I wanted to not only do this non-invasive sampling, but when I wrote for my first grant to go over there, it was NATO. And not only did I propose to have one supervisor, I proposed to have two. So yet again, I had the benefits of working with this guy who was just developing this non-invasive sampling technique. But I worked with this other guy who was a modeler, a computationally amazing person. And I think that strengthened my application. And so that's a long story. I'm going a bit beyond your question, but I just wanted to go overseas because I liked traveling. I'd done it before, and it really opened my eyes to how the world works differently in some places and how people perceive Americans and how they view science. Science is very different in most of Europe. It's controlled more by the lab head. So you have less freedom as a grad student to potentially find something that differs from what the lab director has found before. Your science might be not appreciated or not, you know, there's not as much freedom or encouragement to follow your own path. So just learning those sorts of things, going overseas, and it, you know, it just, travel is one of the best educations. So I would recommend going overseas if you can, and try to find funding, be persistent, and be strategic. Having two supervisors help, because they both are writing strong letters. Hopefully you can help them write their letters. By the way, you learn over time. If you want a strong letter, talk to the person. Don't try not to get a letter from someone of support or recommendation that's good or very good. Try to get one that's excellent, because everyone gets good or very good. And if it's just good, it might actually not help you. And to the you can even, I was really fortunate as a PhD student to get a Fulbright to go to Australia. And I mentioned on the third try, I got it. And that's because it actually was the same lab group and it was the same letter of recommendation, but I read his letter and I realized it could be strengthened. So I wrote the guy and I said, hey, could you change the letter to include how badly Australia needs help with conserving their endangered small marsupials, and how much Australia could benefit from using non-invasive genetic techniques and bottleneck tests? And he understood and agreed, and the letter was stronger, and that was part of why I got it. So just one more helpful story, I think, which is just fortunate, from going abroad and why you should travel. When I was in Australia, I met a theoretical mathematician who was a mathematical genius and a computer coding genius. And I didn't realize it, but I just met him and we were talking about this new marker, microsatellites, pretty new. And all he wanted to know was how they mutated. Was this AC repeat, AC, 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 which is the most common microsatellite in mammals that's used? Did it do a one-step or a two-step or a three-step mutational jump? And it turns out it's mostly one step. But he was simulating data that he couldn't fit to some of his natural populations. And he couldn't figure it out. And we're chatting, French guy, so I especially liked him because I had already been to France as an undergrad. And I said, Jean-Marie, that allele frequency distribution with absence of rare alleles that you can't match in some of your populations is the bottleneck signature. And that's why some of the natural populations you're working on doesn't fit your mutation model, because the population's not in mutation drift equilibrium. It's not in equilibrium, the population crashed. And it's like, wow. So I just was fortunate to connect with and collaborate with a mathematical modeler who we then published really quickly a new kind of test to identify populations that had recently suffered a severe decline. And his math was impeccable and amazing. And then I could just add the application to it because I had all these data sets from small bottleneck populations, real data, and I could simulate some too. And the test worked, and it worked really well. So developed software, published it, and if you want publications, if you want a lot of citations, if you want to advance the field and help a lot of people, you develop software that people can use. And it turns out it's been widely used. And the same with field techniques. If you develop a new technique, non-invasive sampling or something, you can help advance a lot of researchers. Not just, I would advise this, try to make your research impactful, useful beyond your focal species and system. And write that in your papers as well. So choose, research, and write papers that will advance the field and help more people across taxa. And there's certain ways to do that. If you can, I mean, I love giving advice, so I'm going on and on here, but a really important tool in your toolbox, if you're an ecologist, population biologist, community ecologist, population geneticist, is to be able to simulate data. Because if you can simulate data, you can simulate many different species and life histories and scenarios. And if you can do that, then you can run those data through an estimator that everybody wants to use, like abundance. What's the abundance? Like effective size. What's the effective size? What's the connectivity rate? And if you can, and you probably need that if you're using a new cutting edge method for your species, right? You probably need a power analysis. You need to know if it works. And it helps you if you build the simulation model to understand your system, because you got to model it pretty in a realistic way. But then, oh, by the way, you have the simulation system to evaluate the performance of an estimator that you can then apply to all these other species. And suddenly you've advanced understanding of the usefulness of this estimator for an entire field across plants and animals or something. And so the advantages are, you're helping a lot of groups advance the field, not using an estimator that hasn't been thoroughly evaluated and getting the wrong answer. That happens all the time. A new shiny new method comes out. It's not been thoroughly evaluated, but people apply it to their data and then they regret it because their data violated an assumption of the model. So you can advance the field generally. You'll help a lot of people. You'll get a lot of citations. The software, if you develop software, can, be widely used for many years. And it's so cheap compared to producing empirical data in the field yourself, just simulate it. But you can also go grab published data and run it through. So I was talking with someone today who's estimating abundance for multiple carnivores. And there's new abundance estimators. So why not estimate abundance from different estimators on each of these 4 carnivores, but it's only one state. Why not go to another state? Why not grab published data from grizzly bears and, in a really high profile Yellowstone or Glacier National Park. So you become really good at quantitative modeling. It improves your understanding of your system. You can then model many species, you advance the whole field, you get lots of citation, and it's really cheap relative to producing the field data where you got color animals or you produce genotypes. And then a lot of folks will want you to help them analyze their data. So if you're a soft money person, it helps. Or if you have a dry period with no grants, you can pop out papers by simulating data or using other people's data. So become really good. I would say most important for career, there's a lot of things important for career success, but really important is no theory, no data analysis, no data interpretation.
Great. Any other questions from the room, or any questions on Zoom yet? Okay, so we'll move on to a different section. Change pace up here. What's your all-time favorite paper and why? It could be maybe a paper that inspired you early on, or something that the last several years has really changed the way you thought about things. Put you on the spot a little bit.
Yeah, makes me think, I was asked that question in my comprehensive exams. And it's really helpful to be preparing for your comprehensive exams in your defense, because it's one of the few times in your career that you actually read a lot of literature, you have time to read the literature. And I just remember there was this one paper, and this was my answer. And I think the committee loved it. Anyway, a paper came out, and there was this question related to my talk, actually. What's the effect of individual inbreeding and population persistence? And a paper came out, and it showed that mice colonies could be inbred to a certain level, and they did okay. They had a little bit of reduction in persistence or viability and fecundity. And then they just hit this wall, and there was a threshold effect. So it appeared that there could be this threshold effect that if you go past about 0.2 or 0.25 in inbreeding, so 25% of your genome homozygous identical by descent, populations could just crash. I don't know what's known about that today in the wild, but probably is different for each species. But I thought that was just alarming and pretty cool.
Yeah, those thresholds and regime, almost a regime shift at that point. That's scary for conservation.
Yeah, threshold effect. You reach a certain point, and the population is going to decline rapidly, and you might not have time to do anything about it.
Yeah. Cool. Okay. So what areas of research, ecology or otherwise, conservation genetics or otherwise, do you find most exciting right now?
So this is a great interview question. If you're interviewing for a job, you should be prepared to be asked, what's the most exciting question in your field? And ideally, you'll be working on the question, because the next question might be, and what are you doing to address that question? So if you're not addressing that question, so either you have the answer, be what you're doing because you love it, and then your follow-up is, it's what I'm doing. Or, you're ready to write grants to do that because you want to do it, because you realize how important it is. But the most exciting question, I don't know. I switch what excites me fairly often. I'm really interested in the ability to use artificial intelligence, machine learning, pattern recognition algorithms to help managers identify hotspots of spread of invasive species. And so there's a lot of time series data on presence and absence of invasive species across landscapes or riverscapes. I work most in aquatics. And it turns out it's not too difficult with these big databases on presence and absence, thanks to iNaturalist, thanks to, we have a national aquatic invasive species database, and it's USGS run. So there's these sources of where animals are in pixels across the landscape. eBird, so this big data right there on presence absence, and then remote sensing data on environmental variables. What's in this pixel in terms of precipitation, plants, GPP, all these variables, and you can feed that into a machine learning model that looks at a pixel for the organism, identifies the environmental variables and maybe the interactions, and then where they're not, and it learns to recognize pixels with environmental variables that are really high for habitat suitability. And then you can overlay movement of boats, or hunters will move wild pigs around, or domestic pigs, whatever. You can build models that are pretty accurate. I think that's just exciting. And I don't know enough about it, but I would recommend you find out how artificial intelligence how you can collaborate with it and use it to improve your work. I think could be really helpful.
Yeah, the large spatial scale, high resolution studies with these big data sets are going to be important for conservation planning moving forward. So yeah, I agree with you
Yeah,
on
it's
that.
another data analysis,
Yeah.
data interpretation thing. You should learn to do it. And if you can't do it well and don't love it, you can collaborate like I did with that French guy to make it happen. And then I have a brief story. E.O. Wilson, you should read Edward O. Wilson's book, Letters to a Young Scientist, maybe.
Mhm.
Chapter 2 is Don't Be Intimidated by Math. And so you should know Edward E.O. Wilson because he invented the field of island biogeography, for example. And he's just one of the best naturalists. And he didn't coin the term biodiversity, but it made it popular. So his message was to be quantitative, don't be intimidated by math. And when he got hired at Harvard to be their naturalist, he took calculus for the first time because he knew it was important. So he's been sitting in class with students at Harvard learning calculus that he was teaching evolutionary biology to. And so he said, you don't have to be great at math. You have to understand it. Same with coding. You should understand a bit of coding and do a little bit of it. But the way he sort of invented the field of biogeography is he island biogeography, and you should know what that is too. It's the size of the island and distance from the continent predicts the number of species that will be there. He collaborated with a mathematician to do the math. So it wasn't him alone.
Mhm.
And he says in the book, and there's a lot of good advice on how to be successful as a scientist. So again, it's good reading. What was I gonna say? He says, it's more important to have the ideas and the passion in the big picture than it is to be good at math. Because you can find a mathematician. If the idea is good enough, someone's gonna jump on board and do it for you,
Any questions in the room? All right, got another one for you. So are there paradigms in ecology or in conservation genetics that you think are holding us back from progressing or making conceptual breakthroughs?
I don't know. I would be intrigued if the paradigm of natural selection being the only way that populations can rapidly adapt to environmental change wasn't entirely true. And if epigenetic mechanisms could also allow rapid adaptation. It's unlikely to be a widespread important mechanism, but it's possible that in some scenarios during embryonic development, the pattern of gene expression shifted to adapt the organism to warmer temperature or to disease resistance or something. It would be helpful if the DNA doesn't change, but the expression of the gene, because there could be a methyl molecule, methylation upstream of the gene that changes the gene expression. If that change in gene expression is transmitted, is heritable to offspring and grand offspring, then those offspring and grand offspring may be pre-adapted or better adapted to warming or something like that. I just think it'd be really cool if that paradigm of the only way natural selection and adaptation happens is Darwinian, change in frequency of alleles. So, the big A gets more frequent rather than it gets expressed more. And that can happen like that. If it's fixed, and we don't know yet in many species if this happens, but hatchery fish have been shown because hatchery environment's really different than the wild, right? So they have a different gene expression that persists into their sperm, which may persist into fish. This fish goes off into the wild and it has offspring in the wild. What if the wild offspring have that? They might be maladapted to the hatchery. It could happen.
Yeah. I think I can chat about maladaptation of hatchery fish a lot and bore people in the room, but the idea of methylation and epigenetics was starting to get picked up by salmon researchers about 10 years ago or so, but I don't know exactly where they've landed on that. So yeah, I'm right with you on interest in that one.
Yeah, I would say most geneticists suggest, since there's a lack of data, that it's not a thing.
Yeah.
It's at least it's not common and widespread, but it could happen. And in cases where the embryonic development is in such an extremely different environment,
Yeah.
there are data showing that pesticides, starvation in humans, and this radically different environment in hatchery causes different genes to be turned on, and they might not be reset upon gamete development. So they're in the gamete, they might be transmitted to the offspring. And if that happens, you can conceptualize this as an allele getting into nature and then going to high frequency. And it would not be a DNA sequence, it would be a DNA that's methylated, and it's called a methyl allele or a methyl gene. So it could sweep to high frequency, and it might not persist very long, but if it's enough for a few generations of adaptation to occur, it would be a cool thing.
Yeah.
And you might not, I don't know, you might not get easily funded for this because it's sort of viewed as maybe Lamarckian, environmental based adaptation.
Yeah.
But it's such a cool thing. It would change our understanding of evolution. So in terms of basic science, it would be a big deal. But it would have applications to reintroduction of captive animals. So it'd be a really big deal for management. And that's the kind of work you want to try and do. It's things that advance basic understanding and management as well.
Yeah. All right, so we're down to about the last 10 minutes. So if you have any burning questions, don't hold back. Got a few more I can ask. Oh, one in the back there, yeah.
Which one of your questions?
You guys should dream about this often, because I did when I was in early career, but I haven't lately. If I had an unlimited budget, I would try, given my interest and bias, I would try to develop a universal across all mammals or all birds or all vertebrates, cheap genotyping technique that can be used in multiple species and multiple high spots for monitoring, for early detection of population decline and fragmentation. And then maybe have an endowed fund that every year gives thousands of dollars to each of the countries to monitor each of their species and hotspots. Because it could really make a difference. I mean, my travel, I was very fortunate to travel to Ecuador, South Africa, Nepal just a month ago. And it is really concerning, disappointing how limited in funding they are. So our grants are hundreds of thousands of tens of thousands, hundreds of thousands, and maybe millions of dollars, right? So in all these countries, South Africa, one of Africa's richest countries, got a biodiversity hotspot in the south. Their grants are like $1,000 or $10,000, and there's no next gen sequencing, almost no next gen sequencing being used. Not that you need it, but it's really useful for some questions. Even the cheaper methods like GTC, you amplify a bunch of loci and you sequence them. It's $5 or $10 a sample at most. Or RAD captures a method where you sequence a bunch of loci across the genome. They just don't have the money. And I mean, the United States is not a biodiversity hotspot. California is our best. But so the point is, it'd be good for folks to try and help those countries out, and it's a lot of fun. You go there, and I mean, I saw a tiger at 20 feet. I saw that bear I showed the video of charging. It would be really useful in your career if you can afford to do it, to collaborate and help those countries somehow. Write a grant with them for their local funding source, but maybe write a grant in the US that involves helping them out, where you get to go there. Or maybe if you're running a lab someday, you could bring them to your lab and train them.
Great. Any last questions from the audience? All right, so one last one, we talked a bit about your mentors before, but what's your favorite aspect about being a mentor? What have you enjoyed the most about it? Any advice you would give to those hoping to become mentors in the future?
Yeah, I think, so I really enjoy seeing students be successful. It's really rewarding. So I love that. And then a lot of students have taught me a lot. I've learned surprisingly more than I thought I'd ever learn from students. But to be a good mentor, I think you could make an analogy to a good coach in sports. I used to be a sports fanatic. You want to help them be successful in all parts of their life, be good people, never even think of possibly bending rules or fudging data. Work hard. Just be really supporting of them. I just was fortunate, as I mentioned, to have good mentors. And I was warned before I went to work for one of them that he was really demanding. So you got to be demanding. You got to expect excellence, above average work. You got to expect to be pushed. I think people nowadays are a little more soft than we used to be. My advisor would get upset if we were going home he might question us if we're going home for Christmas or Thanksgiving. It's like, what? You got work to do. So people worked harder back then. And I have teenagers, they don't work very hard. Mine work harder than the neighbors, but anyway, to be a good mentor, it can be hard because different students are different. And then you also have to tailor your advice to them. But I would think just being supportive and letting the student know you're going to be there to support them throughout. And tell them when they're screwing up early and tell them how to correct it so that the student can stay on the right path. And if they're falling behind, I hear around here, there's not as strict a deadlines enforced as at some universities, and the University of Montana is a bit like that too. I think it's pretty, and I wasn't forced. I think you should meet with your students weekly until they really don't need it and encourage them to meet deadlines on time. It's a great place to live. People tend to want to stay here and drag on for a 3 or 4 year master's like I think I did. But be better to meet the deadlines and help your students get out the door.
Good advice. All right, got time for one last quick one if anyone has a fast one. Not join me in thanking Gordon for great Q&A. Oh,
Oh, being in Montana, I grew up fishing in Minnesota in Ontario. So I fish more for bass, pike, and muskie. And I have certain muskie lakes where I can catch 10 a day, which is outrageous because muskie's a fish of 10,000 casts. But if I were to fish in rivers, so that's mostly lakes, I would fish the North Fork of Montana because it flows in from Canada. It's beautiful. The fishing's good, and they still have a lot of pure cutthroat trout. And then I caught a grayling there once, which is there. You can get a few species there. If you are a real hardcore fisherman, you might say there's some good brown trout fishing, and you can get cutthroat, brown trout, and maybe even bull trout in some of those rivers. But brown trout's not native, so the North Fork's beautiful though.
that's the North Fork Flathead.
Yeah, so on the western edge of Glacier National Park is this beautiful pristine river that flows in from Canada. And it's a World Heritage Site, biodiversity reserve, and it's just pristine, gorgeous, and wild, and it's a good float. You can just float it and love it. So I'd recommend that.
Great. All right, let's keep going. A round of applause. Thank you for spending some time with us. Oh, and Gordon wanted me to mention he's going to be at West Side Coffee tomorrow morning if folks wanted to meet with him.
Yeah,
his flight's not till later in the day.
Till afternoon. So if anybody wants to show up and chat, I'll be there at 8. 8 to 9 probably, maybe 9:30. West Side Coffee.
So, one more opportunity.